Welcome, dear students, to today's class on parathyroid hormone and the calcium and phosphate regulation in your body. The parathyroid gland located on the posterior surface of thyroid, and the figure is the back view of the thyroid gland, where you can see four different parathyroid glands. It is actually a pair of superior glands and a pair of inferior glands. So the histology, that is the cell types involved in the parathyroid gland, includes principal cells or also known as chief cells and the oxyphil cells. So the principal cells secrete parathyroid hormone, whereas the oxyphil cells, the function is unknown. It has acidophilic cytoplasm, several mitochondria, and pyknotic nuclei. So you'll be wondering what is pyknotic nuclei. So pyknotic nuclei is a nucleus which is small and condensed and is usually seen in apoptotic cells. Respond to low calcium in blood and causes parathyroid hormone secretion from the cells. And the parathyroid hormone acts on osteoclasts via the osteoblasts to increase calcium concentration in blood from the bone. So the PTH acts on kidneys also to increase the calcium concentration in blood, and it also causes the formation of vitamin D, also known as calcitriol, which again increases the intestinal absorption of calcium. So the three major regions where the parathyroid hormone acts is on the bone, the kidneys, and the intestine. So this is the structure of parathyroid hormone, which is synthesized in the chief cells as a pre-pro hormone. So the pre-pro hormone has around 115 amino acids, and it is cleaved intracellularly into proparathyroid hormone, usually in the endoplasmic reticulum. And the proparathyroid hormone, you can see this is the signal sequence or the leader pre-sequence which is cut off. And the proparathyroid hormone is around 90 amino acids. And the proparathyroid hormone packaged into vesicles through the Golgi apparatus, where again it is cleaved to release the active parathyroid hormone, which has 84 amino acids. So this is the pro sequence that is removed. And the PTH can then further be metabolized by the liver into the hormonally active N-terminal peptide and the inactive C-terminal peptide. So the biologically active N-terminal peptide is around 33 amino acids, whereas the inactive C-terminal peptide is the rest of it. So how is the parathyroid hormone secreted? The parathyroid hormone has a half-life of 2 to 4 minutes. So a half-life indicates how fast the hormone is removed from your, or the time taken for 50% of the parathyroid hormone to be removed from your circulation. So in about 2 to 4 minutes, half of the parathyroid hormone that has been released from the parathyroid gland will be. So the secretion of parathyroid hormone is controlled by ionized calcium levels through the presence of calcium sensing receptors or CASR. The calcium sensing receptors, as you can see in the figure, it's a G protein coupled receptor which is found on chief cells, and it can sense fluctuations in extracellular calcium. And these fluctuations causes a conformational change in the receptor, causing its activation or inactivation. So the calcium binds to the CSR and reduces the intracellular cyclic AMP by inhibiting adenine cyclase. So it's a GI receptor, and it also in the process it decreases the parathyroid hormone secretion. So the parathyroid hormone secretion is also controlled by the catecholamine levels and the magnesium levels. So in this figure, you can see that the calcium sensor, in the presence of low calcium, it remains in the inactive form and there is no inhibition of the parathyroid hormone secretion. In the presence of serum calcium, the calcium sensor senses the presence of calcium, binds to the calcium, the receptor receiver binds to the calcium, and as a result, as it's a G protein coupled receptor, it has two different functions. One is it inhibits the adenylate cyclic AMP production and protein kinase A activation. At the same time, it activates the phospholipase C system, which inhibits or which acts as a negative feedback for the parathyroid hormone secretion. So the calcium concentration in the serum is dependent on bone resorption of calcium, kidney reabsorption of calcium, kidney conversion of activated vitamin D or active form of vitamin D, that is 125 dihydroxy vitamin D, and also absorption of calcium in the. So this is the structure of the calcium sensitive receptor. As I said earlier, it has two functions. One is it activates the phospholipase C system so that phosphatidylinositol diphosphate is converted to diacylglycerol and IP3. So diacylglycerol activates protein kinase C, which activates MAP kinase, causes several changes in gene transcription. The IP3 mobilizes the intracellular reserve of calcium from the endoplasmic reticulum, increasing the intracellular calcium concentration, which in effect again activates PKC and produces the same effect. The other function is the G protein inhibitory function, which inhibits adenylate cyclase, thereby decreasing the cyclic AMP concentration, does not allow the PK to get activated, which can cause an inhibition of the MAP kinase. So this is the another depiction of the structure where this is the C-terminal portion, which is sort of in a inverted V shape. And in the presence of calcium, the green dots that you can see is the calcium. And in the presence of calcium, the structure of the C-terminal end of the receptor changes in such a way that it comes together. So this is what I've already mentioned. G protein coupled receptor that activates phospholipase C, increases diacylglycerol and inositol triphosphate as the secondary messengers, also inhibits adenylate cyclase to reduce the cyclic AMP concentrations.